All right, this is my isolating cheek cell DNA lab experiment. And the first thing I'm going to do is make a lysis buffer. And I have a cup labeled lysis buffer. And to do that, I'm going to use 1.5 mLs of Palmolive dishwashing detergent. I have a pipette that is labeled for 1 ml, so there's one. There's the half ml. Recap that. All right, 8.5 mLs of distilled water. Here is my distilled water. Maybe I'll use a new pipette for that. So this is going to be a pain in the butt 8 times. 1, 2, 3, 4, 5.
Tristan, come here. Tristan, get over here.
Eat it.
Mom is doing something. Now be quiet.
6,
Amy, could you type something in?
7,
Come here, I will type it in.
8, and the half. Where's that half hash mark?
Do you want me to turn this on?
A half.
I can turn it on.
Okay, 8.5 mLs of distilled water and 1/4 tablespoon of table salt. What's this? A 1/4 tablespoon table salt. And stir until the salt is dissolved. Stir gently. I might be doing this too rough because it's getting bubbly. All right, I'll let that set. And now I'm going to make the Pro-Tease solution. I have a cup labeled Pro-Tease solution. I need 10 mLs of distilled water. And we'll use this pipette again. So 1, 2, 3, 4, 5, 6, 7, 8, 9. All right, 10 of distilled water. And 1/8 of a teaspoon of meat tenderizer. I don't have an 1/8 teaspoon, so I'm gonna eyeball a half of this 1/4 teaspoon. And hopefully that will work. And that looks like half to me. All right, and stir until the tenderizer is dissolved. Pro-T solution. All right, now I need to chew on the inside of my cheeks and mouth for 30 seconds. I'm just gonna count in my head. All right, now I have 3 mLs of tap water. I'm going to put in this cup. I'm going to rinse my mouth out with it for another 30 seconds. That doesn't look like a lot. Gross. All right, I spit that water back out, and I'm going to transfer that water to a 15 ml cap tube. This is the 15 ml cap tube. This is my spit cheek water. Super gross. Okay. All right, using a clean plastic pipette, add 2 mLs of lysis buffer to your tube. So this is the lysis buffer that has been sitting here. Make sure I will use a new pipette. 2 mLs of lysis. There's 1. There's 2. Recap. All right, I'm going to gently invert this 5 times. 2, 3, 4, 5. I'm not sure if I see anything changing, or I can see stuff, maybe something sort of suspended in the liquid, but I'm going to put that back in my cheap cell cup. All right, using a clean plastic pipette, add 100 UL of Pro-T solution to the 15 ml tube containing your cell extract. The second line on the pipette is the 100. Here is a new clean pipette. Here is my Pro-T solution. And the meat tenderizer looks like it's totally dissolved. And it says the second line. The second line on the pipette looks like it's labeled 0.5 ml. All right. This is going into my 15 ml tube. And gently invert it 5 times. 1, 2, 3, 4, 5. All right, we're gonna let this sit for 10 minutes. Go ahead and pause. 10 minutes have gone by, and my 15 ml tube has laid here undisturbed for the full 10 minutes, and I have just retrieved a bottle of isopropyl alcohol from the freezer. I'm going to fill this 15 ml tube with up to 10 mLs of cold alcohol. I'm going to grab another clean pipette. holding the tube at a 45-degree angle and gently allowing the alcohol to go down the inside of the tube, not disturbing the contents too much underneath. Oops, wrong cap. All right, I'm going to be letting this tube sit for another 5 minutes undisturbed. I can see two definite layers, and in the midst of the clear layer up top is sort of like a, well, it looks like a collection of bubbles. I'm not sure if that's what I'm supposed to be seeing, but that's what I see. So I'm going to put this back in my cup, and it's going to sit here for another 5 minutes. Okay, it's been 5 minutes, and my tube has been sitting here at room temperature, undisturbed. As I look at it, I can see there's definitely a bottom cloudy layer and there is a top clear layer. And in the midst, sort of maybe rising from the bottom is sort of a, it looks kind of like a wispy cloud with a collection of bubbles, you know, at the top. Sort of, you know, floating there in the solution, but also maybe like attached to the bottom, like it's sort of rising up from the bottom. This collection of bubbles is rising up from the bottom, and they're all sort of attached to each other. So I'm going to invert this tube again. 5 times. So here we go. 1, 2, 3, 4, 5. All right, so according to this lab sheet, my DNA has been isolated in this solution. The salt and the alcohol made the DNA cells sort of clump up here together. And as I look inside of that tube, that wispy, filmy that was moving up from the bottom has now sort of consolidated, I guess, a bit more. And it looks more white and stringy than it did earlier. Before I mixed it, it looked more wispy and definitely like a collection of bubbles, and this is more like still wispy but cloudy-ish wispy, not as many bubbles. It's more, like I said, just kind of like a wispy cloud, and apparently that's how you separate DNA.